Okay, so today's lecture is on multifactorial traits. Multi means many, so there are many factors that can contribute to a trait or to a characteristic. We know that in real world human beings, we do not have a lot of Mendelian genes. It means Mendelism was all about one trait being controlled by a single gene. So it was a very utopian kind of concept where one gene is entirely in total control of the expression of one characteristic, like, you know, the T, the capital T and the small t, they were controlling the height of the plant. And every gene had these two distinct versions of it, or two distinct alleles. So those are known as Mendelian genes or monogenic genes. So that is a trait that is controlled by a single gene. But in real world, few, if any, genes actually act alone. So most of the traits are controlled by multiple genes, polygenic, okay? And irrespective of whether it's a Mendelian gene or a polygenic inheritance, there are so many other environmental factors that can also influence the expression, the phenotypic expression of the genes. So both Mendelian and polygenes could be multifactorial. They are really complicated. It's a complicated network of influences, not just a genetic influence, but environmental as well. So this is a nature versus nurture controversy going on. So single gene traits, that means those characters which are controlled by a single gene, are actually qualitative. That means they have either 100% effect or zero effect. For example, if you had a gene for the capital T version, okay, whatever for there, then you have a really tall plant. Otherwise, there is nothing. Or if that gene is absent, you don't see anything, any of that trait. That trait is totally absent. But in case of polygenic traits, you have a range of expression. For example, the gene for height, and that's why we have such a range of heights in human beings. You know, it can go from anywhere, adult human being can go from anywhere from 3 feet all the way up to 7 feet. So this is the range. Or skin color. We have all shades of skin color. We have from the lightest light to the darkest dark. We have different colors of brown, different light skinned and dark skin. So this is a continuously varying phenotype or shades of gray. So these are known as quantitative traits. So there are qualitative versus quantitative. Qualitative means single gene, quantitative means polygene. And those genes, that means those DNA sequences that contribute to these polygenic traits, we give them a fancy name. We call them quantitative trait loci. Loci is a plural, locus would be singular. So genes are located in positions, they are known as loci or locus. So the DNA sequences in those regions which are controlling, which are, you know, going for trait that are polygenic, those DNA sequences are known as QTLs. These are some examples of polygenic traits: height, skin color, body weight, fingerprint patterns, behavioral traits. All of these are not simple inheritance, not controlled by one gene. So let's take an example here. This is the example of height. Remember, Mendel's law always holds true at the genetic constitution level. So genotypically, it would still be true. So when you make a Punnett square, you see this is exactly following Mendel's laws. But their phenotypic expression is the one that differs, okay? So they do not produce the single gene phenotypic ratios, and there are so many reasons for that. Because sometimes effect of genes could be additive. We call it synergistic effects. That means one gene has this effect. Let's say it makes the plant tall or it makes the plant short. But there could be a second gene and a third gene and a fourth gene which present in certain combination, or which present or absent could be modifying, could be adding or subtracting to or from the effect of the height. So the frequency of distribution in case of these type of genes is actually forms a bell-shaped curve. This is a continuously varying phenotype. That means you will see some plants, some individuals who are really short in height, then you see individuals increasing in height, and then the tallest individuals, and again, the height goes down. Another very interesting thing is fingerprint patterns, okay? And the branch of science where fingerprints are studied is known as dermatoglyphics. This is very commonly used in forensics as well. What are fingerprints? Fingerprints are, if you look at your fingers, you see the skin there is not very smooth. There are ridges and grooves. That means ups and downs. They are very small, microscopic. If you take a magnifying glass, you'll see them much better. And you know that every individual has unique fingerprints. And that is something very interesting, because identical twins who are genetically identical, so they have the exact same genetic composition. So you would assume that at least identical twins would have the same fingerprints, because fingerprints that are being controlled by genes, they have the exact same copy of genes. However, it is seen that even identical twins do not have the same fingerprint patterns. And you know, fingerprints on our fingers, these ridges that you see, they can be arranged in different patterns. They have loops, we have arches, we have walls. And the reason that they are different in every individual and even in identical twins is that, of course, genes play a major role in determining your fingerprints. But in addition to it, it is prenatal contact. Prenatal contact means when the baby is still unborn, and it is usually before the 20th week of gestation. So halfway through pregnancy. Pregnancy is total of 40 weeks of gestation. You know, even before 20 weeks of gestation, as the fetus is growing, the fetus is touching the inside of the bag in which it is growing, right? So it is touching parts of its own body. It is touching, you know, the umbilical cord. It is touching the inside of the mom's uterus. And as it is touching it, the skin there is still so soft, it is impressionable. So when it is touching it, that touch, that contact is actually changing the fingerprints, the friction ridges on the fingers of that unborn baby. And that is why two identical twins growing inside, you know, the mom's uterus at the same time, genetically the fingerprints were supposed to be the same. But since the babies are moving their limbs, they are moving their hands, and they're touching each other, they're touching themselves, they're touching the inside of the mom's uterus, and that is why the fingerprints become different. So the fingerprint patterns are something that is controlled by, you see, multifactorial traits. It's not just genes. There could be more than one gene involved. And there is this maternal environment, the environment inside the uterus when they were growing as a fetus. Height is a very common example. The difference in height between two sets of students, it is not only just genetic. Of course, your genes play a very, very important role, but nutrition, health, overall health also plays a very important role. It was seen that during the Irish famine, when people did not get enough food to eat, the average height of the population decreased, okay, because the bones, due to the absence of calcium in their diet, they really did not grow as long as their ancestors were. So GWAS studies, that means genome-wide association studies, have shown that at least 50 genes, is not just a single gene, at least 50 genes contribute to height, okay? Another multifactorial trait that we see in human populations is skin color. And we all know from common sense, we have all heard of melanin. Melanin is the name of a pigment. Biologically, it's a polymer. That means it's made up of many small units. And this pigment protects us. The more melanin we have, the darker the skin color. So it protects us against, the melanin is a protective agent against the harmful effects of ultraviolet radiation. Now, you will be surprised to know that if you take human beings who have very different skin colors, if you take a little bit of their skin, per unit area of skin, human beings actually have the same number of melanocytes. That means those cells that make melanin. So, two individuals with different skin colors would have the same number of melanocytes per unit. That means density of melanocytes is the same, but the difference is in the number of melanosomes that they make. Melanosomes are these little bubble-like structures, little vesicles, into which melanin is put inside. It's like melanin is the protein. It is packaged inside this box called melanosome, and then the melanosome is released, and then the melanosome starts moving out, and then the melanin will do its job. So it is difference in the number of melanosomes, in the size of the melanosomes, in the density of the melanosomes, that creates a difference in the skin color. You see in this figure here, these are twin sisters, but you see the difference in the skin color. And this is because of, if you see this bell-shaped curve, here only three genes have been shown. So let's say gene 1 is indicated by the alphabet A, gene B, is the second, this allele B is the second gene, and C. So you see the different possible combinations of these three genes. As soon as a person has a dominant A, B, and C, that means at least one capital A, one capital B, and one capital C, the skin color gets darker. And the more the number of recessive alleles, so an individual with all small a's, small b's, and small c's have the lightest skin color. So all possible combinations of these three genes would give rise to all this wide range of skin color. Now, in world, people usually associate skin color with race, but that is a total a misnomer. Why? Because the definition of race, if you base it largely on skin color, it is actually socially made. This is not a biological concept, or this is not even biologically correct, because skin color is not a reliable indicator of ancestry. Nowadays, so many companies have come up. You just have to provide some spit sample, and they will give you your ancestry, how much percentage of your ancestry comes from let's say American Indian or African ancestry or East Asian ancestry or European ancestry. So many people are very surprised to find that phenotypically what they look, they might be very light skinned, they might have blue eyes, blonde hair, and yet their ancestry shows a lot of maybe Asian, component, or even, African component. So the basis of race on skin color is biologically, it doesn't hold true. Investigating multifactorial traits. So there are certain terms that we use when geneticists work on these multifactorial traits, and they're working on human populations, especially now with the COVID-19 situation going on. If you listen to the news, you might hear these terms very commonly. So it's good to know, what do these terms mean? A very common term is incidence. Another term is prevalence. What is the difference between incidence and prevalence? Incidence is the rate at which a certain event occurs, while prevalence is the proportion or number of individuals who have that particular trait at a particular time. That means, in simple words, prevalence would mean how widespread a disease is in a population. You can think about the COVID-19. What is the prevalence of COVID-19? While incidence would refer to new cases of the disease in the population in a year. So it hasn't been a year yet, but we can still calculate, okay, in these many months, these are the new cases of COVID-19 that would give you your incidence. While the entire calculation, the entire count would be the prevalence. And another term is empiric risk. Empiric risk measures the chance, the probability that a trait or a character will recur based on incidence, based on this year. So these are some of the terms that you would commonly find getting used in population genetics. Another multifactorial trait is this cleft lip, which is a very common genetic disorder. It can be cured by a minor surgery. And this runs in families. So cleft lip would be more likely in a person who has a relative with this condition. Now, another term that you would use even in this class in some of the subsequent lectures is the H factor, or the heritability factor. Heritability means the proportion of the phenotypic variation for a trait in population due to genetic differences. That means we are trying to understand, when we look at a population and we see a certain trait, and we are trying to understand how much of it is contributed by the genes, while how much of it is contributed by the environment. So how do we quantify this? A quantitative measure, a test is done, and that gives you your H factor, heritability factor, and that tells you, and we will learn about this when we do population genetics. We have to learn about Hardy-Weinberg law to understand this. But for now, just memorize this definition and understand the big picture of what heritability, or H, means. Using some genetic calculation in a population, we can predict how many individuals in that population are expected to show that particular trait. So that gives you your expected number. Okay, so theoretically or ideally, if everything goes right, then this is the number of individuals that should show this trait. That is your expected number. And then what you do is you look at the population and actually count the number of individuals who show the trait. So you get your observed number. So the heritability calculation is comparing observed versus expected. Okay, so you see how many you had predicted should have occurred and how many are actually occurring. And then the maximum value for heritability is a 1.0, that means 100%. So heritability equals 1.0 for a trait which is varying only due to genetic reasons. So if it is only genetically controlled, no environmental influence at all, it's like a very ideal situation. Okay, no environmental interference, only genetic, then that particular trait would have a heritability of 1.0. And the lower you see the heritability value going from 1.0, that means the more the environmental influence. Now, coefficients of relatedness. When we see hereditary traits, when we want to see, look at this disorder, we want to see how much is being influenced by the environment. How do geneticists study it? They study not only your parents, they would like to study the entire family. As many family members information that you can provide is better. Why? Because all of these members are related to us genetically. So if you're thinking about, what is the relation between the parent to a child genetically, you see it is called a primary degree of relationship. Okay, so 50% of the genes would be shared. While if you want to see the relationship between an uncle or an aunt to the niece or the nephew, this is called a secondary relationship, 25% shared. Grandparent to a child is also a secondary relationship, 25% of the genes shared. First cousin to first cousin is a third degree of relationship. This is 12.5% of the genes will get shared. And this is how when doctors diagnose a particular child with a problem that is supposedly genetic, and they send you to a genetic counselor, the genetic counselor sits down and let's think that if this is the particular child or this is the particular individual in question, Tim, so then the geneticist would draw a pedigree chart like this. Okay, that will show all the primary, the secondary, and the tertiary relatives, and then the geneticist would start marking down who all had the disease in order to understand or in order to confirm what the doctor is suspecting. Adopted individuals. With common sense, we know that if it's an adopted child, of course, all the traits, they are not going to have any genetic component. It's all environmental. So any similarity that you see between an adopted child and adoptive parents always reflects environmental influences. There's no genetic factor there. However, if an adopted child can go back and trace and find out his or her biological parents, then that child who hasn't lived with the biological parents has lived with the adoptive parents. Whatever similarity between between that child and the biological parents are purely genetic, because that child did not get any environmental influence by watching his or her biological parents. So these are very interesting cases which are used for genetic studies, where they study this child who was adopted very early on in life. So this child did not really see the biological parents, but then they compare the traits that the child has that are common between the biological parents, between the adoptive parents and the child, because this provides you information on nature and nurture, on heredity and environment, how these are contributing. Twin studies, twins have always fascinated geneticists. Why? Because it gives them the option of seeing identical, semi-identical and fraternal twins, because you know how much of the genetic material is common between them. Now, two measures are used, the two definitions, concordance and discordance. Okay, concordance means it measures the frequency of expression of a particular trait in both members of a twin pair. It could be a monozygotic or a dizygotic twin. So if you have identical twins, which are monozygotic twins, okay, they are more concordant. That means they're more close, genetically more close to each other. While if you look at the trait in fraternal twins, they are genetically more different from each other. So they are more discordant. So concordance would be higher for identical, that is monozygotic twins, than dizygotic or fraternal twins. So separating genetic and environmental influences. Again, you see twin studies being used. And if you have monozygotic twins, if you can find enough monozygotic twins to study, you know that the genotype is exactly the same, okay? And they might have shared environment if they are living in the same household, if they are living in the same family. Dizygotic, they have the same environment, same household, same family, same parents, but only 50% of the genes. Now, a second layer of complication is if you can find twins who were raised apart. Okay, they were separated very early on in childhood. So that means they have a genotype which is shared. They could be identical or fraternal, but the environment became very different. So now you can study the traits in there, while adopted individuals provide you another dimension to this study, where they are sharing only the environmental influence, not the genes. Now in these studies, SNPs play a very important role. SNPs, if you've forgotten from the previous chapter, SNPs means single nucleotide polymorphisms. That means these are changes in single bases of the DNA. This is the DNA double helix in one individual. Imagine this is the DNA double helix in another individual. And remember, there are the nitrogenous bases here and there. So if you see, here is an A, here is an A, T, T. Well, this pair, which is in red, for this individual, this is in G, and for this individual, this is in T. This is a mutation in a single base. So we call all of these mutations. When do we call it a single nucleotide polymorphisms? If you see that this is the most common type, that most people in a population show AGT, okay? But very few people are showing ATT, then you count how many people are showing ATT. And if at least 1% of the population show an ATT, then it will be considered a single nucleotide polymorphism. If less than 1% of the population show the ATT, then it will not be considered a SNP. It will be called a random mutation. Okay, so it has to be that 1% is the cutoff. That is the threshold. So we see SNPs, we use SNPs to compare groups of population. GWAS studies, genome-wide association studies, also depend, a lot on SNPs. And if you go, there are certain databases. If you go to UCSC, University of California, Santa Cruz, they have a genome browser, they have a database, and this is a big HapMap project. We discussed this in the last class, that it is not possible to do high throughput studies where you have thousands of individuals. It's not possible to study every single gene individually. Instead, what do high throughput studies do is they have haplotype blocks of gene that behave in the same way, that get inherited together, they stick together. So those are haplotype blocks. And in those haplotype blocks, we just choose a representative SNP that is called a tag SNP. And we just study that tag SNP. And by just studying 1 or 2 tag SNPs in big blocks, big chunks of genes, we can predict about that entire chunk. So it reduces the effort, the money, the time that you have to put in to study all the genes by just looking at hap maps. You just look at haplotype blocks with their tag SNPs. So what are SNPs used for? SNPs can be used for personalized medicine, to know if this particular individual, or first of all, to find out the mechanism of why this group of people have the disease while the other group does not have. So what we do is we take people with a certain disorder. Let's say, these people have cancer while these people do not have cancer. So this is your case group, this is your control group. And we get DNA we extract DNA from these individuals. We also extract DNA. So this is our case or patient DNA. This is our non-patient DNA. And then we look for SNPs in these DNA. Okay, these are molecular techniques. We do that. And then we identify what are the SNPs that are common in these group of individuals, and those SNPs which are different in this group of individuals. Now this gives you a clue of which genetic constitution makes people more susceptible to cancer. It's because these people had this kind of genetic sequence that made them have cancer, while this group of people had the genetic constitution different, so they did not get cancer, or they did not get any other disease or any particular trait. This is a very commonly used strategy that geneticists use to compare, first to come up with what are the things that are going wrong in these people with a certain disease. And this is a stepwise approach where we start with a lot of SNPs, and then we slowly focus on, we bring it down, number of SNPs to very, very specific genes. It's like a funnel. So when we have a, huge population and we want to cover all the genes because we don't want to miss anything, we start with, let's say, about 500,000 tag SNPs. Okay, we have a case control study. We have 800 cases of cancer, 800 controls. And we start with about or more than 500,000 tag SNPs. Now remember, they're just tag SNPs. That means there are more SNPs there. And then we carry out the studies. We see what are the differences. We reduce it to, we bring it down to 25,000 SNPs. Another follow-up study going deeper into those SNPs, bring it down to 1,500. Finally, narrow it down to, it comes out to be 25 specific genes. And then this one would be a really good finding, might get published in one of the very good journals, because now we have narrowed it down from 500,000 tag SNPs, brought it down to very, very specific 12 particular genes, and these are the genes that are responsible. So now if people have to do any kind of genetic engineering, gene therapy, or some drugs that can actually reverse the effect, the proteins that are not being made because of that mutation here that caused the SNPs, a lot of the drugs that you use in cancer, specifically some type of cancers, actually, when they find that there was a particular SNP that was mainly responsible for that disease, they insert medicines in the body, and those medicines can go and reverse the effect of the abnormal protein that is being created. So the medicine discovery or medicine designing, the drug designing was possible because initially somebody did this. So a lot of treatments depend on these things first. These are the baby steps to finding out cures. So unless you do this, how do you design a medicine? So study designs in genetic studies, in heredity studies, can be two types. I have been telling you about case control study, very common, where we have a group of cases versus a group of controls, a disease versus a non-disease, and then we see the SNP differences. Another type of study is called a cohort study. In cohort study, researchers will recruit a large number of individuals, volunteers who will volunteer for the study, and then an effect is seen over time. Okay, for example, I work on a study on diabetes, and I have a group of individuals being studied for several years now, and we provide them dietary interventions, we provide them medicines, and we have field workers who go there on a regular basis and are collecting information from them, if they, if those individuals are following the directives that have been given to them, if they're taking the medicines, if they're doing exercise as they have been recommended to. So those are known as cohort studies. Another type of study design is the affected sibling pair strategy. Means you take siblings, you take brothers, sisters, and you then scan the genome of the siblings for SNPs that are shared. So, in a sibling, one of them might have a disease, the other one does not. So then you take the DNA and you compare what happened, why this change, why does this particular, a sibling have this while this one doesn't have it? Is there any difference in the genetic constitution that made this particular brother more susceptible? Homozygosity mapping. Homozygosity mapping is usually performed on families that are consanguineous. In certain cultures, in certain countries, it is very common amongst close relatives to get married. Now, this is genetically not very recommended. The reason being, when people marry in close relatives, the recessive mutations, they get a higher chance, there's a higher chance of those recessive mutants to come up in double doses, because people in the same family may have the same recessive mutations. So if you are marrying outside of, if you're marrying somebody who's not related to you, there's a very low chance that you will also end up with another individual who has the same recessive mutation. But there is a high chance if you marry a person in the same family that you end up with another person who also has that same mutation. So let's say a small r in both individuals, and that when they get married, their offspring, then that two small r's come together, and there you go, double dose of a recessive mutation, then that particular trait gets expressed. So the children in this case are more likely to inherit two copies of the mutation. Now, the Jiva studies, which have, which are still very common, and still, people go for these in order to understand a lot of the diseases. There are certain limitations when we do these high throughput studies. These are mainly epidemiological or, studies that provide us with the trend, but not the causes. Yes, we know this particular genetic variation or SNP pattern is making the person or making these group of individuals more susceptible, but it is not actually shedding a lot of light on the mechanistic basis, not the cause. Also, you have to be very careful when you're recruiting your group of patients or controls. You cannot let bias come in, okay? It has to be very unbiased, because if there is bias, then you're prejudiced against, okay, let me include this person. Oh no, this person looks very sick, I am not going to include this person. No, then your recruitment is biased. Body weight. Height and weight, both of these are continuously varying traits, but it has been seen for several years of research that height, although both are controlled by genes and environment, height has more genetic influence while body weight has more environmental influence. So that is why, although genetically it might not have been made that way, but 30% of all adults in this country are obese. Okay, 35% are overweight, and nowadays, each time you go to the doctor, the doctors, in the office, they make you step on the weighing machine, on the height, and they give you your BMI, they give you your weight, they let you know if you're overweight or obese. So this is all environmental influence. Why? Because our lives have become very sedimentary, very sedentary, not sedimentary, but sedentary. We don't move around that much. Our cars are parked in our garage, and then we just go from there. Then when we go to office, we try to park closest to where the building is. So it is a lot of this sedentary, not very active lifestyle. So body weight, what about the genetic component? What are the genes? Are there genes? Yes, of course, there are genes that are involved that have effect on hunger and how your metabolism works. And not just metabolism varies from person to person, but it also varies in the same person in different ages, right? So as you get older, your metabolism slows down. So you might be eating the exact same food that you were eating 10 years ago, but then 10 years ago you did not put on weight, but when you were at 25, and then when you are 35, then the same diet, you might start putting on weight. It's just because now your body cannot metabolize that fat. It is storing it up because metabolism rate has slowed down. Some of the common associated proteins are leptin and ghrelin. Leptin is a hormone, it's a protein hormone which is produced by adipocytes, that means your fat cells. And this hormone, leptin and ghrelin, they are antagonistic, they act in opposite manner. So leptin actually goes to the brain, and then it acts on some of your neurons, and then it tells the body that you're not hungry anymore. It decreases the appetite. So you get the feeling of, okay, I'm full. Okay, I don't want to eat. While ghrelin is a peptide hormone, that means it's a protein-based hormone which is produced in the stomach, and this responds to the stomach's needs. Whenever the stomach, digestive system lets it know that, okay, it's time to get some food, time to get some energy. And then the ghrelin, it increases appetite. So this hormone goes and it stimulates your neurons. Your nervous system starts telling your body that you're feeling hungry, so you eat. So it has to be a proper balance between leptin and ghrelin that keeps the body weight, in an optimal range. And we are done with multifactorial traits. Any questions for me? Now remember, you have your discussion session today. Make sure you go over, whatever questions that you might have, how to solve genetic problems. You have quizzes coming up. Your homework assignment 1 is due today. You also have midterm coming up next week. So start preparing for those. Do not procrastinate, okay? If you joined in late, make sure you write your name in the chat box before you leave. Okay, I guess there are no questions, so I'll end the meeting. I'll see you tomorrow.